Welcome back. Today on The Explainer, we're diving straight into a fascinating mystery right at the crossroads of physics, biology, and medicine. We're talking about biomaterials. You know, those incredible engineered materials designed to heal us, replace damaged tissue, or just seamlessly interact with our biology. But here's the really cool part. We're going to decode the secret language these materials actually use to communicate with us, and look at the wild tools we use to listen in.
So picture this. You're standing in a biomedical lab. It's late at night. It's super quiet, except for that low hum of the ventilation. And sitting right there on a stainless steel table is, well, something that looks completely ordinary. Maybe it's a thin membrane, or a clear hydrogel, or just some simple polymer film meant to heal damaged tissue. To anyone just walking off the street, it's literally just a piece of plastic-looking stuff, right? But to a scientist? Oh, that material is completely alive with hidden information.
But that brings up a massive challenge, doesn't it? How do we see what the naked eye simply can't? I mean, how on earth do we know if that clear little hydrogel is actually releasing medicine, or if that scaffold is successfully supporting living cells? Well, to figure that out, we turn to a trusty device called a spectrophotometer. Now I know it's really easy to look at this thing and just see a boring beige box sitting on a lab bench. But trust me, it is so much more than that. It's practically a magical translator. It takes the totally silent interactions between photons and molecules and translates them into a language of biological function that we can actually read and understand.
Section 1. Seeing the Invisible World.
Okay, let's dive right in and look at the absolute foundation of how this translation works. And it all starts with light. Light is our ultimate messenger here. Think about it. When light strikes matter, only three things can possibly happen. It gets absorbed, it gets transmitted right through, or it gets reflected back. Now, biomaterials interact with light because the molecules inside them are absolutely packed with electrons and chemical bonds. And those microscopic little components, they eagerly respond to electromagnetic energy. So, if we see a material absorbing very specific wavelengths of light really strongly, it's basically shouting at us that something specific inside its molecular structure is grabbing onto that exact light.
Section 2. The Language of Light.
Figuring out this language of light was a total game changer. It literally shifted the entire field of biology. Early scientists made this brilliant, albeit kind of simple observation. More molecules just mean more absorbed light. And that beautifully simple idea evolved into the Beer-Lambert law. Absorbance equals molar absorptivity times concentration times the optical path length. Now, why is this a big deal? Because this is the exact moment biology turned into measurable math. Instead of a researcher just squinting and vaguely guessing, hey, I think this scaffold has some collagen in it, this one equation allowed them to definitively state, no, this scaffold contains exactly 2.3 milligrams per milliliter of collagen. It was the massive leap from just passively observing things to actually quantifying them.
Section 3. Decoding molecules and nanoparticles.
So how do we actually put this to work? Well, our first go-to tool is UV visible spectroscopicometry, which operates in this very specific sweet spot, usually between 200 and 800 nanometers. Why that range? Simply because a ton of crucial biological molecules happen to absorb light right there. For example, aromatic amino acids and proteins love UV light around 280 nanometers, and DNA absorbs it right around 260. The best part? Scientists can evaluate a drug or a protein without destroying the sample. Light essentially becomes non-invasive biology. Now, what's really interesting about this slide is the contrast in how we investigate these materials. We've got UV vis showing us electronic transitions, but what if researchers want to dig deeper into the material's identity? Enter FTIR, or Fourier transform infrared spectroscopy. Instead of just looking at electronic absorption, FTIR lets us literally tune in to molecular bonds vibrating. Think of chemical bonds like tiny little springs. They're constantly stretching, bending, and twisting. Because different bonds absorb infrared light differently, FTIR gives every single biomaterial its own totally unique molecular fingerprint. How cool is that?
And honestly, this interaction between light and matter creates some of the most stunning phenomena in all of science, especially when we shrink things way, way down to the nanoscale. Quick question, did you know that gold nanoparticles actually appear red? Or that silver nanoparticles look kind of yellow-brown? No way, right? You'd expect gold to look, well, gold, but it's not because of some added chemical pigment. It actually happens because the electrons inside these insanely tiny particles are collectively oscillating or vibrating in sync with the incoming light.
This exact phenomenon is known as surface plasmon resonance. It's a bit of a mouthful, but because of this effect, every specific size of a nanoparticle produces a completely different, unique absorption peak on our machines. What that means for us is that UV-Vis spectroscopy can tell us right away about the formation, the stability, and the exact concentration of these particles. Literally, just a tiny microscopic shift in that peak can reveal massive structural clues about what is happening down at the nanoscale. It's kind of like reading tea leaves, but with physics.
Section 4. Tracking life and time.
Now, we have to remember, biomaterials aren't just dead, static objects sitting on a microscope slide somewhere. They're specifically engineered to interact with the human body, which means we have to introduce a whole new variable to the mix: time. See, a lot of biomaterials are actually designed to intentionally disappear. Drug carriers dissolve, hydrogels slowly degrade, and scaffolds get resorbed right into the body. So to track all this action, researchers use our trusted spectrophotometer to measure the absorbance repeatedly. We're talking over hours, days, or sometimes even weeks. Then, jumping back to our good friend the Beer-Lambert law, they calculate the exact concentration being released at every single time point. Suddenly, the biomaterial isn't just a thing. It becomes this dynamic, evolving system, letting scientists map out these beautifully detailed release curves and degradation timelines.
And as these materials degrade, they're making room for something incredibly important: living tissue. But when scientists first see a synthetic scaffold with actual cells, the suspense in the lab builds immediately. I mean, the very first, absolutely crucial question they have to ask is, are the cells actually alive in there?
And of course, the questions don't just stop there. They need to know, are those cells proliferating? Are they multiplying and growing? Basically, are they actually doing what they're supposed to be doing? To figure all of that out, we rely on tissue engineering assays. Things with names like MTT, LMR Blue, XTT, and crystal violet staining. Here's the trick. Living cells will actively metabolize, or basically eat and process, these specific dyes. And as the cells process the dye, you get these massive, very visible color changes happening right there in the solution. By popping that into our machine and measuring those changes in absorbance, the health and vitality of the cells become perfectly quantifiable. Just like that, the scaffold is no longer just a piece of inert matter. We can mathematically prove, for sure, that it has become a living, breathing environment.
Section 5. AI and optical biosensing.
Let's move forward and see how this builds into the real future of medicine, right where biomaterials collide with modern diagnostics. Today, biomaterials are increasingly being used as optical biosensors. Imagine, for instance, taking enzymes and immobilizing them on a polymer just to detect glucose. It reacts, creates a colored product, and boom, the absorbance level tells you the exact blood sugar level. Pretty clever, right? But these days, we definitely don't just stop at reading basic spectra. We're taking these incredibly complex spectral signatures and feeding them straight into machine learning and deep learning algorithms. That single spectrum suddenly turns into massive, actionable data. We're literally using AI-driven analysis of light to classify cancerous tissue or instantly spot invisible bacterial contamination. It's an absolute game changer.
Section 6. The Quantum Physics Bridge.
Now, to truly appreciate how any of this is even possible, from counting those tiny cells to running advanced AI diagnostics, we have to zoom all the way in. I'm talking down to the absolute foundational layer of reality itself. Because underneath all of this tracking, underneath the algorithms and the AI, lies pure quantum physics. Think about it. Electrons are occupying these very discrete energy levels, and molecules possess their own specific vibrational states. Meanwhile, incoming photons of light are carrying highly specific amounts of energy, defined by that classic equation you see there, E equals h nu. If a photon's energy perfectly matches the molecular energy differences of our biomaterial, that is when absorption occurs. So here's the really wild takeaway. Those simple peaks and squiggly graphs we observe on our lab machines, they are actually just the quantum shadows of electron behavior. And that really brings us full circle. What starts as a tired scientist just wanting to study an ordinary-looking membrane suddenly reveals this massive, beautiful intersection of optics, molecular biology, nanotechnology, and quantum mechanics. The spectrophotometer isn't just some lab instrument, it's a literal bridge connecting physics and biology. Every single spectrum we decode tells a unique story. A story of structure, a story of interaction, a story of healing, a story hidden entirely in light. So it kind of makes you wonder, what invisible stories do you think the everyday materials around you are telling right now? Keep learning, stay focused, and I'll see you in the next explainer.